Most animals and other organisms have built-in clocks in their brains that regulate the timing of biological processes and daily behavior. These clocks are known as circadian rhythms. They allow maintenance of these processes and behaviors relative to the 24-hour day-night cycle in nature. Although these rhythms are maintained by the individual organisms, their length does vary somewhat individually. Therefore, they must, either continually or repeatedly, be reset to synchronize with nature's cycle. In order to maintain synchronization, entrainment, to 24 hours, external factors must play some role. The reason why entrainment occurs in humans is because each individual's circadian rhythm is longer than 24 hours. Majority of population, or shorter than 24 hours. Minority of population. Of the various factors that influence this entrainment, light exposure to the eyes is the strongest. Melatonin plays a large role in effects of light on circadian rhythms. When an organism is exposed to a light stimulus, the hormone melatonin is suppressed, or prevented from being secreted by the pineal gland. Demonstrated effects. All of the mechanisms of light-affected entrainment are not yet fully known. However, numerous studies have demonstrated the effectiveness of light entrainment to the day-night cycle. Studies have shown that the timing of exposure to light influences entrainment, as seen on the phase response curve for light for a given species. In diurnal, day-active species, exposure to light soon after awakening advances the circadian rhythm, whereas exposure before sleeping delays the rhythm. An advance means that the individual will tend to wake up earlier on the following day. A delay, caused by light exposure before sleeping, means that the individual will tend to wake up later on the following day. The length of light exposure influences entrainment. Longer exposures have a greater effect than shorter exposures. Consistent light exposure has a greater effect than intermittent exposure. In rats, constant light eventually disrupts the cycle to the point that memory and stress coping may be impaired. The intensity and the wavelength of light influence entrainment. Dim light can affect entrainment relative to darkness. Brighter light is more effective than dim light. In humans, a lower intensity short wavelength, blue-violet, light appears to be equally effective as a higher intensity of white light. Internal pathways and mechanisms. Light first passes into a mammal's system through the retina, then takes one of two paths. The light either gets collected by rod cells and cone cells before being transmitted to the retinal ganglion cells, RGCs, or it is directly collected by these RGCs. The RGCs use the photopigment melanopsin to absorb the light energy. Specifically, this class of RGCs being discussed is referred to as intrinsically photosensitive, which just means they are sensitive to light. There are 5 known types of intrinsically photosensitive retinal ganglion cells. IPRGCs, M1, M2, M3, M4, and M5. These connect to amacrine cells in the inner plexiform layer of the retina. Ultimately, via this retino-hypothalamic tract, RHT, the suprachiasmatic nucleus, SCN, of the hypothalamus receives light information from these IPRCGs. The core region of the SCN houses the majority of light-sensitive neurons. From here, signals must reach the rest of the SCN in order for circadian shifts, or entrainment, to occur. There are specific genes that determine the regulation of circadian rhythm in conjunction with light. When light activates NMDA receptors in the SCN, clock gene expression in the region is altered and the SCN is reset. And this is how entrainment occurs. Genes also involved with entrainment OPER1 and PER2. Some important structures directly impacted by the light-sleep relationship are the superior colliculus pretectal area and the ventrolateral preoptic nucleus. Secondary effects. While light has direct effects on circadian rhythm, there are indirect effects seen across studies. Seasonal affective disorder creates a model in which decreased day length during autumn and winter increases depressive symptoms. A shift in the circadian phase response curve creates a connection between the amount of light in a day length, and depressive symptoms in this disorder. Light seems to have therapeutic antidepressant effects when an organism is exposed to it at appropriate times during the circadian rhythm, regulating the sleep-wake cycle. In addition to mood, learning and memory become impaired when the circadian system shifts due to light stimuli, which can be seen in studies modeling jet lag and shift work situations. Frontal and parietal lobe areas involved in working memory have been implicated in melanopsin responses to light information. In response to light exposure, alertness levels can increase as a result of suppression of melatonin secretion. A linear relationship has been found between alerting effects of light and activation in the posterior hypothalamus. Disruption of circadian rhythm as a result of light also produces changes in metabolism. Other factors. Although many researchers consider light to be the strongest cue for entrainment, it is not the only factor acting on circadian rhythms. Other factors may enhance or decrease the effectiveness of entrainment. For instance, exercise and other physical activity, when coupled with light exposure, results in a somewhat stronger entrainment response. Other factors such as music and properly timed administration of the neurohormone melatonin have shown similar effects. Numerous other factors affect entrainment as well. These include feeding schedules, temperature, pharmacology, locomotor stimuli, social interaction, sexual stimuli and stress. Treatment. Dark therapy before bedtime, where light that triggers melanopsin receptors is filtered out with eyewear, is considered an effective method to promote earlier production of melatonin and improve sleep. See also chronobiology, circadian advantage, circadian clock, circadian oscillator, circadian rhythm disorders, electronic media and sleep, light therapy, scotobiology.